Welcome back, Mindsetters. I hope you guys are ready for the session for Learn Extra for grade 11s. We're doing life science. So guys, I hope you're ready for the session. Make sure you get your pens, pads out and ready to make notes, because Kathy's about to tell us what's going to happen today. So awesome, Kathy, what are we doing today?
Awesome, Ty. Guys, this is your session. I have nothing that I still have to go through with you. Just a reminder that your exams are probably going to be on circulation, all right? So transport systems, which would be the heart and the pulmonary system and all the different types of systems that we've got. So you'd have that. You've got excretion, which is the whole kidney thing. And then, of course, you've got the viruses, the bacteria, the algae, and the fungi, which we have to go through as well. So you need to let us know what it is that you want me to go through. All right. Ty, I think we may have some questions if you want to push refresh, and let's have a look.
No, definitely, I will do that.
So
actually, while you make your way there, I'm going to check
you
here.
Have a look, and then
I will make sure that
you're going to read it to me.
Yes, I'm going to tell these guys, make sure you guys get on the page, get chatting to me. You know the link, www.facebook.com/learnextra. Send in your questions. Whatever subject you guys really need help on, make sure you speak to me so I can get that to Kathy so I can cover it today. So, while I'm on that point, let me actually see what we have here. Yes, we have one already from Mashatsi. He wanted to find out. If we could please do the nephron.
Oh, okay. I'm just making a list here of what you people need to know, and then we've got viruses.
And then, because Jameson actually wanted to find out what if we could do bacteria as well.
So you just keep, I can do one thing at a time though.
No problem.
And we've got bacteria, viruses, fungi, and we have protists. Okay, people, if we have a look here, this is what you need to know. You should have covered by now transported in animals, and that would be circulation. So that's the heart and all the blood vessels and double circulation, blah, blah, blah. And then we have excretion, which is the whole kidney and the waste removal process. Then we have viruses and bacteria and fungi and protists. So those are the sections that we do with you. Now the first one was, if I will go through the nephron. Okay. Let's do the nephron. Okay. Now what you've got to understand is the nephron is the single filtering unit in the kidney. So it'd be like having a block of flats, and you see this entire big building, but the building's actually made up of a whole bunch of little flats in little rows. All right, your nephron is the structure that does the actual filtering process. Now, if we look at a kidney, right, you'll have two very distinct regions that you can make out of a kidney. The outer region is called the cortex. And the middle region, because it's in the middle, is called the medulla. And what's really important is the fact that your nephron filters in or cuts into both regions. So you've got the little nephron sits here. Look, this is not according to scale. And it'll go wiggle woggle, and then it drops down into the medulla, and then back wiggle woggle, and then back down into the medulla. And this here would have another nephron and another nephron, and all these little nephrons joining in, and this becomes your collecting tubule. And your collecting tubules make up the pyramids, which are made up of papilla, and your lesser calyx, your greater calyx, and then they all join in like that, and eventually the urine passes out from the pelvis and then down the ureter. Okay, so this is what we have. What we are interested in is this little structure here, and that is the nephron. Now, if we look at a nephron, the nephron's made up of different parts or components. we have the first part is like a little bulby thing. And you listen to the words I'm using here. It's a little ball. It's like a little bulby thing. And then it goes wiggle, woggle, and it drops right down. And it goes up again, and it goes wiggle, woggle, and down. Okay, and you're saying, oh, for heaven's sake, Kathy, what's wiggle, woggle? Wiggle, woggle is when a tube goes wiggle, woggle. Okay, and when you have to draw this in an exam, wiggle, woggle. So here we go. And we go wiggle, woggle, down, and then the tube gets thin. And it is very, very thin all the way around here. Then it goes big, wide, and long, and we'll have, let's put another collecting tubule. So we'll have a collecting tubule, another nephron attached there, and another nephron attached here, and another nephron attached here. Okay, so if we look at this region here is called the medulla. Okay, I'm going to take this out. This part is the medulla region of the kidney, and this part is the cortex, just like I showed you over here. So cortex and the medulla. And if we look at the cortex is a dark maroon color, and it sort of looks granularly if you touch it. It's got like a lumpy texture. Whereas the medulla is beautifully smooth, and it's a light pink because it's made up of all these tubes. It's made up of tubes and not these little balls. Now, what is this little ball? This little ball has a blood flow, and the blood comes in here, and you've got a capillary network, and then the blood exits. And that one's wider, and this one's narrower. The fact that this part is wider and that one's narrower means that we have pressure. Now, people, whenever we want to create pressure, whenever we take a fluid and we put it from a larger tube into a narrower tube. And the minute it goes from a larger to a narrower tube, pressure will build up. It's a simple thing like, if you go, when we have our break, I want you to go to the kitchen. I want you to put your finger at the tap, and I want you to open the tap. And you're going to find that if you're blocking the hole to that tap, you're going to be able to spray water all over the show. The minute you pull your finger away and the gap is larger, the water will flow easily. Then stick your finger there again and close that gap by putting pressure on it, and you're going to find the water will have more pressure and it will spray. That is exactly what happens here. If at this point here, this is the afferent arteriole, and the afferent arteriole, just think of A for arrive. The afferent arteriole is larger than the efferent arteriole. Now, the fact that it's an arteriole tells you that it's gone from an artery. The artery is split up into arterioles, and arterioles are now going to make capillary networks. Except that here, the arteriole comes into a structure, and it exits, and it's still an arteriole. So how? Why? Because it hasn't gone through the organ. It's only once it starts to go through the tissue in the organ does it form a capillary network. And then when the capillaries start joining up again, it will form a venule and then a vein. So we say, right, this is E for exit. Okay? And this, the afferent being larger than the efferent, that creates pressure. And it creates pressure for our first process, and the first process is called pressure filtration. So your pressure filtration, okay, is caused because the afferent arteriole is larger than the efferent. And it's bringing blood that is full of wastes, but it's also blood that is full of nutrients. And it's blood that's full of oxygen. Okay, so the kidney cells themselves need oxygen. That's number one. Number two, they need nutrients so that they can do their job. Okay, but what's their job? Their job is to remove the wastes and make sure that by filtering all the blood, it picks out the waste, and we weed that out, and it takes all the good things back. It's no good that all the blood is filtered and then we end up urinating out all our good things. We want to keep those good things. We only want the wastes to pass out. So, afferent, larger than efferent, and that's process number one. I'm going to put here 1. And that process number 1 is pressure filtration. This structure here is called the Bowman's capsule. Okay? And that Bowman's capsule and this little structure here is called the glomerulus. And the Bowman's capsule plus the glomerulus is called the Malpighian body. Okay, so we just look at this little structure here. We have the Bowman's capsule. I'm going to do it. We have the Bowman's capsule and the glomerulus, with the afferent arteriole is larger than the efferent arteriole. And the glomerulus plus the Bowman's capsule is called the Malpighian body. Okay, so these two together, Malpighian body. And the first process that we have that's important is pressure filtration. Now we've got something else, and I'm going to make it in lime green. Sitting here around the inside surface of the Bowman's capsule, we have little structures. Those little green things are not aliens. They are called podocytes. Now, if I look at the inner lining of the Bowman's capsule, the Bowman's capsule is made up of cells, one layer of cells, because remember we're going to have absorption and diffusion here. It's made up of one layer of cells, and they are squamous cells. Okay? And then sitting between the glomerulus, remember here's the glomerulus with all its capillaries, and the afferent arteriole, and we have the efferent arteriole, which is slightly thinner, which creates pressure for pressure filtration. Here's the glomerulus. Between the glomerulus capillary and this epithelial, squamous epithelium, there's a little thing that looks like a crocodile clip. So it sits like this. Okay, and then you'll have another one there. And those little things are called podocytes. So they sit here between, around the at least around the squamous epithelium, between the endothelium of the capillary and the epithelial cell. So what do? They fit literally like this. So there are very tiny gaps there. So anything that comes out of the capillary, all right, any substance that comes out of the capillary, so glucose and amino acids and water and all the good things and the bad things that come through here from the capillary into the Bowman's capsule as filtrate, it has to pass through those slit pores. These are called slit pores. And as it passes through those split pores, they don't allow anything large to pass through. That's their job. And we call that process number 2, which is ultrafiltration. And what causes the ultrafiltration? The podocytes that sit here on the inner layer of the Bowman's capsule. And what happens is they don't allow, these podocytes will not allow proteins and your red blood cells and fats to pass through into this Bowman's capsule. And why would that be important? Because remember, whatever goes through that Bowman's capsule and sits in that Bowman's capsule as filtrate, the good things must be taken back into the body. And only the bad things and a bit of water must pass out. So what do the podocytes do? They say, if we lose red blood cells, we're not going to be able to get them back. And guess what? We do need fat in the body, because remember, fat is stored energy. And proteins, oh my gosh, we're not going to throw proteins away. Definitely not. If you have any amino acid, not proteins, amino acids in your urine, it means your liver's not deaminating. If you have proteins in your urine, it means that your kidney cells are damaged. The actual kidney is damaged, because otherwise your podocytes are going to be filtering out your red blood cells and your proteins and your fats. Okay, people? So you never ever find proteins or red blood cells or fats in urine. If you do, there's a big, big problem. All right. We are supposed to reabsorb all the glucose and all the amino acids. If we have glucose in our urine, it means you're a diabetic. It is an indication of note that you are diabetic, because the kidneys are designed to absorb all the glucose back. Amino acids, whoa, that means your liver's not working. And if your liver's not working properly, you're basically weeing out all your proteins, or your amino acids, you have a huge problem. And they'll take you to hospital and deal with it.
All right.
So our clock's not working. And
no,
I just felt you staring at me
and I was giving you that mental telepathy of...
Absolutely, because we're so attuned, aren't we?
Exactly. So, on that note, guys, make sure you don't go anywhere, you stick around. And yes, I'm still giving away these packs. Make sure you get on the page, get chatting to me. Whatever you guys need help on or need us to look into, make sure you get us on the page, because I know a lot of you are saying that you're writing tomorrow. So make sure you chat to us so we can help you guys out. But for now, we're going to see you after this break. Welcome back, Mindsetters. I hope you had a nice little break and you guys did whatever you need to do. You went to the bathroom so you don't have to fidget and move around and wiggle, so you can focus and pay attention, and you got your pens and pads out and you guys are making notes, because I know that a lot of you are writing tests tomorrow. So make sure you're paying attention. And if there's sections that you critically need help on, post them on the page and we'll see if we can cover them in this session. And guys, I cannot stress enough, I still need to get rid of these. So make sure you keep chatting to me. But for now, I'm going to hand over to Kathy. Kathy, take it away.
Thanks. I see all your questions. I want to cry, because if I had 5 hours and we could sit and do 5 hours, I would be happy and I would feel that I have given you everything I can. Okay. I'm not supposed to spot for you, but if I was your teacher, I would give you a comparison between viruses and bacteria and protists. Okay, and what have I left out? Viruses and fungi, and give you a comparison. And I told you when we went through it, draw up a comparison table and learn that table, so you know what they are, where they are, what they look like, how they move, how they eat, how they reproduce, and that's it. And do it as a comparison. When you compare things, you remember them. All right, so let's get through our kidney here. So if we look at the Bowman's capsule, or in this case, the Bowman's capsule plus the glomerulus, which gives us our Malpighian body, we have two processes that occur there. One is pressure filtration, because the efferent arteriole is larger than the efferent, and we have ultrafiltration because of the podocytes. Okay? So remember the podocytes, and they sit on the inner layer of the Bowman's capsule. Now we've got everything that's been filtered into here, except the red blood cells and fats and proteins, because the red blood cells, fats, and proteins are going to go where? They're going to stay in this efferent tubule here, or efferent arteriole, and they are now going to form the capillary network, they're going to form the capillary network that's going to now reabsorb all this stuff. Now we're going to go on to this part here. And that part is called the proximal convoluted tubule. So as we come from our Bowman's capsule, we're now going to go to the proximal convoluted tubule. Okay, so I'm doing this part here. And that part goes like this, and then it starts to do this. Why? Because this here is the cortex. It's still the area in the cortex, and there it starts to dip in the medulla. The fact that it goes from a wider to a narrower tube, that immediately tells us there is pressure. Okay, now sitting in this tube here, sitting in this Bowman's capsule, we have the good guys. So we got the good. We have the bad. We have water. Okay, that's what's sitting in there. Good, what good? Amino acids, glucose, fructose, galactose. Those are the good things. Okay, vitamins, mineral salts. We want them back. So what do we need to do? We need to make sure that we have a way of reabsorbing. Now look at the word, reabsorbing all the good things back into the blood. We want the bad things to pass through all the tubes into the collecting tubule and out through the ureter and into the bladder so we can wee it out. We don't want it. We want to excrete it. All right. And water, well, that's fine, except that whatever water goes in here, okay, that water has to be divided between sweating and cooling the body down, okay, and urine to flush out the wastes. So the body has to decide, hold on, it's a hot day. Can I afford to make 10 liters of urine when I'm only drinking 1? No. All right, and if I'm only drinking 4 liters, guess what? I need 3 liters to cool me down, so make sure that the urine is going to be very concentrated. Or it's a cold day, and I'm not sweating a lot, and I'm taking in 4 liters. So guess what? I don't need to cool the body down, so I've got to make sure I wee out. 3.5 liters must go, otherwise I'm going to start swelling. The body needs to do that, and this process here is done with which hormone? Come, I want you to tell me which hormone it is. And while you're thinking, I'll give you the letters. It's ADH. And what hormone? I want you to think what the hormone's proper name is. Now, while we are here in the cortex area, this region here, okay, we have all the little goblet cells that sit along here. Again, just one layer of cells all the way around here. There is a lot of active transport. And that active transport causes and starts the reabsorption. So our next process in our row of processes, this will be process 3, is going to be reabsorption. And that reabsorption is by active transport. So what happens is the glucose goes from here and it is actively pumped out of the tubule. And amino acids are actively pumped out of the tubules. Out, get out, go, go, go. And out they go, and what do? They now make a higher concentration here, a higher water potential on this side, and there is a lower water potential in the tubule. So, sorry, I've got this wrong around. Makes a lower water potential outside and a higher water potential in the tubule. So what happens? The water follows. And this accounts for 80% of the water and the amino acids and the glucose and all the good things. 80% of it gets reabsorbed back into the blood over here because of active transport. Okay, now what happens? We sit with a fluid or a filtrate passing down the tubule and it's now heading to the medulla. And you think, why? Well, first of all, the tube narrows, and then it goes into this area here. So there's the division between the cortex and the medulla, and our tubule's done this now. It goes here, it gets narrower, and then it does that. This part of that nephron is called the loop of Henle. And Henle is written with a capital letter, by the way, people, so it's the loop of Henle. And what it does is, this is the descending tubule, that's the ascending tubule. And we now have our filtrate here, and that filtrate has already had 80% of the water and the mineral salts and the vitamins and the glucose and the amino acids is back in the blood. So this filtrate now drops into the medulla. And the tube narrows to put pressure. And when it gets into here, we have the fifth process, process number 4, and that process here is called the sodium pump. And what happens is that the cells lining the loop of Henle literally pump sodium into the surrounding tissue. And now we have sodium, and guess what? Chlorine follows. Because remember, sodium chloride love each other. And you must remember that from grade 9 if you don't do physical science now. Remember it from grade 9? Sodium, chlorine love each other. And sodium chloride makes salt. It's the stuff we put in our food. So it pumps out the sodium. The chlorine follows. What do? We lower, we lower the water potential. So what happens? The water says, whoa! And the water molecules follow by osmosis. Out goes the water. So now we've taken a whole bunch of water out more. So that's process 4, which is the sodium pump. Now this fluid starts to climb uphill again. And when it gets to the top there, so it's now just gone into there's cortex and medulla. It's just dipped down. Okay, now it starts to dip up. And here it comes. So it goes wiggle, woggle, and here it starts to go down again. So it goes zhoops, wiggle, woggle, and now we're going to have our collecting tubules coming in here from different, each one of these will be another nephron. So this area here is the distal convoluted tubule. And what happens at the distal convoluted tubule? Well, we've now gone into we've been in the cortex, gone down into the medulla, where the sodium pump was, and that was because of a hormone called aldosterone. And now we're heading up again, and we're back in the cortex. You think, what? Why are we getting cortex medulla? Because in the medulla, we have aldosterone. And remember, aldosterone is produced by the adrenal gland. And here, in this part, we have antidiuretic hormone. Remember I said you must think of what ADH stands for? It's an anti-urinating hormone. It's an anti-weeing hormone. It wants to keep water in your body. So if I've got lots of ADH, I'll keep lots of water in the body. Little bit of ADH, less water in the body. So antidiuretic hormone makes sure that whatever little bit of water that we started off with is now reabsorbed. Okay? Why? It makes the cells of the distal convoluted tubule, and this is our 5th process, they become more permeable to water. And why are they more permeable to water? Because of antidiuretic hormone. All right, so water now is again reabsorbed. By now, we're sitting with very concentrated urine because we've now taken in all the the good stuff was back in the blood, and the bad stuff has to pass out. You need to think of the kidneys filtering process like Your mom comes home and says she's won the lotto. She's just won 10 million. And you are all going shopping. Okay? Now, you go to your cupboards and think, whoa, I'm going to go into my cupboard and I'm going to just chuck everything out and I'm going to just buy brand new stuff. Okay? So what do? You go to your closet, okay, the blood, and you chuck all the clothes out onto the floor. It's now sitting in the Bowman's capsule. The floor is your Bowman's capsule. And then you see your favorite pair of jeans. I'm not throwing that away. And you fold and you put it back in your cupboard. And you see your absolute favorite jersey. It's that jersey that you've had for the last 5 years, that every time your heart's been broken, that's the one you wear, and that's the one you sleep with. Okay. And you see your, I don't know, Ty's hat that he loves so much.
Which is always on my head.
He will never, I mean, even if he won the lotto himself and he had 10 million, he would still have his hat because he loves his hat. So these are all the things you take back, okay, that go back into the closet. The closet is the blood. So all the good things are taken back and they folded and put nicely back into the closet. But what are you left with? You're left with everything else. And that everything else then you put into bags, which is the waste. And you go and you take it out of the house and you give it to people that need it. So people, that is, if you think of that analogy, that's what the kidneys do. So we now go to our fifth process, which is ADH, makes the tubules more permeable to water. And then we get to our collecting tubules. And your collecting tubules are very special because what happens here is, now, apart from collecting from the various nephrons, in this area here, what we have above this area sort of there of the collecting tubules, we have, it's what we call, what's termed tubular, and this is process 6, tubular secretion. And anything in the blood that doesn't want to be there, anything extra that's bad, will literally be pumped into the distal convoluted tubule and the collecting tubules as tubular secretion. And that is why you have more urea here in the tubular secretion than you had in the Bowman's capsule. Okay, that's the whole process. So, process 1 is in the Malpighian body. We have a pressure filtration because the afferent is larger than the efferent. Then we have ultrafiltration because of the podocytes and the slit pores to prevent red blood cells and fats and proteins from passing into the filtrate. Okay, then we get to the distal, and at the distal convoluted tubule, we have selective reabsorption. Then it drops down into the loop of Henle, where we have the sodium pump. Then the liquid pushes up in from the loop of Henle into the distal convoluted tubule, where we have antidiuretic hormone, causing the cells to be more permeable to water. And then we have the collecting tubule, where we have tubular secretion. This would be, for me, a perfect essay. If not, they can give you a section of the diagram and then ask questions on it. But it's a lovely essay to say, discuss the various filtering processes and reabsorption processes of the nephron of the kidney, or the filtering unit of the kidney, which is the nephron. Just last thing I want to do with you, how are we going for time this time?
I think before their brains actually explode.
You want to take a quick break?
Yes.
Cool.
All right, so guys, I'm seeing a lot of you are saying like, okay, want to touch on this subject, want to touch on this. Guys, I'm going to do my best to try and see which one is the one that you guys most want to find out about. And guys, I'll make sure I get that to Kathy. But for now, we're going to see you after this break. Welcome back, Mindsetters. I hope you had a nice little break and you got something for yourself to drink. Nice and warm. You got your blankets because I got myself some tea because I'm freezing. But, you know, we're going to talk about more about the body system and all those kinds of things. So, guys, make sure you get those questions in on the page so I can get them to Kathy so she can help you guys out. And I'm going to hand straight back to Kathy. Kathy, take it away.
I feel so frustrated because I've got, you guys are like asking so many things and I'm thinking, how can I choose one thing over something else or over something else? But I'm trying, really. So we've got about 10-15 minutes left. A lot of questions have come through for circulation. Something that I would ask if I was a teacher in a test or an exam, I would ask you to do a comparison. So to compare the types of blood vessels. Okay, which I'm going to do very quickly and very briefly. Okay, so remember you've got three different types of blood vessels. Okay, you're going to have your arteries, and I haven't put these lines straight, so let's just make, I need space. So that's why those old style where you've got four green blackboard, not green, they're green boards, four green boards in a row in a class, that's why they're so lekker, because you can go from one to the next day. We actually need to get 4 of these in a row. But okay, here we go, people. You've got your arteries, and you've got veins, okay, and you have capillaries. Okay, so arteries are always larger than veins, which are smaller, and capillaries are the smallest. Okay, I just want to show you here. It goes heart. From the heart, you have arteries. Arteries split up into arterioles. Okay, your arterioles split up into capillary networks. Right? Then your capillary network starts to join up, and it joins up into venules. And your venules all join up into veins. And the veins then go into the heart. Okay? At the same time, your heart is going to pump to the lungs, and it comes back to the heart. And that's your double circulation. So, if we look at the heart, arteries split up into arterioles, split up into your capillary network. They join up to form venules, and they join up to form veins, and the blood goes back to the heart. From the heart, it'll go to the lungs, from the lungs back to the heart, and then to all the parts of the body again by this process. So, how do we get the blood to go where it's got to go? In our blood vessels. Why? Because we have a closed system. Arteries are larger, the veins are slightly smaller, and the capillaries are the smallest. If we look at the artery, it's got a smaller lumen, but it's got a thicker muscle layer. Okay? Whereas with your veins, have a larger lumen. Okay? The overall structure is smaller, and it has thinner muscles. Thinner muscle layer. Okay, your capillaries are just one single layer, a layer of endothelium. Endothelium is the layer, it's one single layer of cells, is what a capillary looks like. Okay. Your veins are going to transport blood. If we look at, where am I here? Here. So your arteries are always going to transport blood away from the heart. Veins always transport blood to the heart. Okay, and your capillaries are always in between. So they are just small little structures that link arteries and veins. Okay? Let me just take this off here. Okay, with arteries, there's always pressure. So there's huge pressure. Okay, veins, there is a lack of pressure. And in capillaries, there's slight pressure. And it will only be slight pressure as the arteries are now going into arterioles, which then go into your, let's do it this way, which then goes into your capillary network. That's your venule side, that's your arterial side. It's always slightly larger, and it creates pressure, and your little cells will be in between here. Okay, so there's pressure, here there isn't pressure, and this is why we will not, there are no valves in your arteries, whereas in your veins we have semi-lunar valves. And clearly in your capillaries there are no valves. Now, John has posted on the web, okay, on Facebook, he's posted comparisons for you. Please learn that comparison, okay? Very, very important. Now, Ty, what was the other heart question that's been, everyone's been asking?
It was more about the circulation, probably, I think, from what I'm gathering, it's circulation.
Hang on, it was the valves. It was
yes,
about the valves.
Valves.
Okay, cool. All right.
Then circulation, sorry.
Now, if we look at the valves of the heart, okay, here we go. And this is really, it's as simple as I can make it. Okay? If we look at the heart, this is what the heart looks like. It's got cavity, 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 and cavity. Okay? No, this is not a mask. What's that movie series where they walk around with a scythe and they kill people and they've got this kind of mask thing?
Scary movie.
Scary movie type. It's not a scary movie mask. Okay. This is just my drawing of the the heart. Okay. So this is it. The heart has 4 chambers. The 2 on the top are atrium. So this would be an atrium, and this is an atrium, and this is a ventricle, and this is a ventricle. Okay? Now, what do we have? The blood comes in. The blood has to now go from here. It goes into this ventricle here. This is the left side. This is the right side. Okay? The blood comes in. Inferior and superior venae cava goes into the atrium. It goes into the ventricle. From the ventricle, it's got to go to the lungs. Okay? This blood is deoxygenated. It's got no oxygen. It's come back, but it's got nutrients. So it's come from the digestive system. And it's got waste that's come from every single organ in the body and every single cell in the body. So it goes to the lungs. Then the blood has to come, the yellow will be oxygenated blood, comes from the lungs, and it comes in here into the atrium. From the atrium it goes to the ventricle. From the ventricle it pumps out to all the parts of the body via the aorta. And here it's going to everywhere. Okay? This blood is going to be oxygenated. But it's still carrying the same waste that we had in the blood that went to the lungs. Only difference is oxygenated versus deoxygenated. Okay, now, in order to stop this blood from flowing backwards and to create this pressure, we have valves. And we've got two lots of valves. We've got the bi- and tricuspid valve. Let's get green. We've got the bicuspid valve and the tricuspid valves here. So your cuspid valves sit between your atrium and your ventricle. So between these two, we have our cuspids. Okay, those are your cuspid valves. Now the blood that's passing from here to the lungs and out there to the atria. When the ventricle relaxes, we don't want that blood to flow back in the heart, because if that blood flows back in the heart, there's no space for the new blood to come in. So you don't want that. You've got to make sure that that blood doesn't flow back. So what do we have? Here in the pulmonary artery which leaves, so in the pulmonary artery which leaves the heart to take blood to the lungs, which is deoxygenated, over here we have a semilunar valve, and in the aorta, because when this ventricle stops contracting, that blood's got no more pressure. That blood's going to start running backwards. So we have a semilunar valve there. Okay, so we've got cuspids and we've got semilunars. You've got tri and bicuspid. Bi means 2, tri means 3. And because your right is, so we write with our right hand, remember it this way. In the right side we have the tricuspid, and then the left side we have the bicuspid. But they're the cuspid valves. Or you can call them the atrioventricular valves. Why? Because they're in between the atrium and the ventricle. So you've got your atrioventricular valves. Those are the two valves between the atrium and the ventricle, left and right side. And then you have your semilunar valves, which work with pressure. Now, for your bi- and tricuspid valves, what happens? Atrium here, ventricles there. So the blood goes psh, into the ventricle. Now the ventricle contracts. As that ventricle contracts, the blood is being pushed from the ventricle up into the atrium, because it's a I'm pushing and it squishes the blood. So the blood now comes whooshing up into the atrium. We don't want it back in the atrium. So what happens? The valves close. So your bicuspid or tricuspid, or if you want to call it the atrioventricular valves here, they slam shut. Dwah! That is the lub sound of your heartbeat. Now there's pressure, and that blood is being pushed by the ventricles all the way. This blood goes to the lungs, this blood goes out the aorta to all the parts of the body. There's immense pressure. But then the ventricle relaxes. Okay, you can't have this poor little thing squishing away for all eternity. It's got to relax. So what does it do? It pushes like anything for 0.3 seconds and then says, oh, I'm done now, and it relaxes. When the ventricles relax, the blood that's flowing up to the lungs and out the aorta, that blood's going shh, and the next thing there's no more pressure. So what happens? The blood will now start to flow backwards. And as it does, we've got that these semilunar valves have been open. Now what happens? Semilunar valves in the pulmonary artery and in the aorta, what do? The blood now, there's no more pressure, so the blood starts to just chill and come back. As it does that, it closes the semilunar valve. So we have cuspid valves, semilunar valves. Cuspid valves between the atrium and the ventricle, semilunar valves in the pulmonary artery carrying blood to the lungs, and your aorta taking it to the rest of the body. There we've got the semilunars, which work as due to a lack of pressure. Okay, and sure, that's about people, that's it. So remember, your cuspid valves, tri- and bicuspid valves, they whack shut because of pressure from the ventricles, so that the blood can go out via the pulmonary artery and the aorta. Lack of pressure, the blood doesn't flow back, it just causes the semilunar to close. And in your lymphatic system, in all your lymph vessels, you have your semilunars as well to prevent the blood from the lymph fluid from flowing backwards. And that's all yours, Ty.
Wow, that was so much information.
Who's got the books?
I'm going to make sure that I look at up and down the page and make sure I find the guys who've been posting the most. And guys, you get the books if you post the most. Simple math in life science. But besides that, guys, make sure you come back on after the 16th of July, because we'll be back. No, 16th of June.
Just hang on. Good luck for your exams.
Good luck. And guys, goodbye. We'll see you next time.